Alright Mustang, so here's a quick tutorial on how to complete your monster synthesis project. So we all know that DNA is a set of instructions for making proteins, which then build and maintain a living thing. So in this project, you're going to create a monster by taking DNA instructions, transcribing them into mRNA, translating them into the amino acids of a protein, and then determining what that protein is going to do for the monster. Okay, because that's really how it works. DNA has the instructions, has to be rewritten into RNA, specifically mRNA, read by the ribosome, turned into a protein, and then that protein is either going to help build the organism or maintain it on a daily basis. Okay, so on this example here, so the very first one, it says number 1, gene for hair color. So you're going to have a portion of DNA that's going to determine what color your monster's hair is going to be. So what you need to do is look at the other handout here, and you go to the part where it says number 1, hair color. So it matches number 1 gene for hair color, number 1 DNA strand for hair color. And you're going to pick one of these 4 strands, not 2, not 3, just only one of these. So I'm going to pick sequence number 3 right there. So I chose sequence number 3. So I'm going to write down sequence number 3 right here. And then I'm going to copy exactly what it says for sequence number 3 right here where it says DNA. Because these are the DNA instructions that are going to determine what color my monster's hair is going to be. So I'm going to write down TAC GGG CCC AAA ATT. Okay, so there's my DNA sequence that's going to determine what color my monster's hair is by making a protein in a very specific way. So next I gotta transcribe it. Remember the scribe part. Transcribe means to rewrite the DNA into mRNA. So T becomes an A. A, when it gets rewritten into mRNA, it can't become a U. I'm a T, sorry. It has to become a U, because remember there's no T in mRNA, so it has to be a U. So A becomes U. C, that's a G. That's C, C, C. G, G, G. T, oops. U, don't make my mistakes. U, U. And U, A, A. And don't forget your good habits. After every 3, draw a line. And the reason we draw a line, remember, is because those are the codons. Each codon codes for an amino acid. So that's one codon right there. There's another codon there. That's going to tell me what amino acid to put down to build the protein. Okay, so then I go AUG. I use my chart. So if you have this chart here on the bottom of the class set or the separate handout that I gave you. So the very first one is AUG. So I go A, U, find the box where they meet, and the last one is G, so methionine. So I put down MET. Remember, all proteins are going to start with that. That's the very first amino acid that starts off our proteins. The next one is CCC. So I go C, C, find the box where it meets. Next one is C, it's proline, PRO. Don't forget your little dashes to show that the amino acids that we're putting down here are connecting together through covalent bonds to actually build the protein. Next one is GGG. So I go G, G, find the box where they meet, and the last one is G. So I put gly for glycine. U, 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 U. Be careful on this one because this box has PHE for phenylalanine and LEU for leucine. And you want to make sure you do the right thing. So it's U, U, this is the box where they meet. You have to look at this side here, U, bring it across, PHE. And finally, UAA, so UAA is stop. So that tells the ribosome, stop building the protein, don't add any new amino acids, you're done. Alright, next thing we need to do is find out what this protein codes for. That's where the chart that's connected to the back of the class set comes in. So when you look at this little chart here, what we're going to look for is because every single protein starts with methionine, MET for the start, and then it has a stop codon at the end. We have to basically ignore those at this point. We're just going to look at these three in the middle. Now, a real protein is going to have hundreds or thousands of amino acids connecting together to build it. Here in this example, it's only using 3. That's not very realistic, but just know that in real life, though, a real protein would have hundreds or thousands of amino acids to build it. All right, so proglyphy. So we look at here, we look for proglyphy. So proglyphy, that's going to build a protein, or this is going to be a protein that causes my monster to be pink and purple. So right here on this line where it says physical appearance protein, I'm going to put pink and purple. Okay, so that's my first one. I got my hair color. Next thing is the gene for eye color. So if we move it there, we have the gene for eye color next. So I'm going to look at this, and right here, go to number 2, pick a DNA strand for eye color. I'm going to pick one of these sequences here. So I'm going to pick sequence number 2. So I'm going to write sequence number 2 here. I'm going to copy it straight exactly. If you don't copy it exactly, you're going to get the wrong answer. So make sure you copy it exactly. So TACATA CGC GTA, and finally ACT. Okay, so I copied my DNA sequence. Now I need to transcribe it. Transcribe means to rewrite it into mRNA. So T goes to A. A is rewritten into a U, not a T, because there are no T's, no thymines in RNA, just uracil. C, that becomes a G. Good habits, I'm going to put a line. UAU line. GCG line. CAU line. UGA. Okay, now I need to transcribe it. So AUG, we know is going to transcribe into methionine. I'm sorry, translate. It's going to translate into methionine. UAU. So I go UAU. It's TYR. Next one is GCG. So I go GCG is ALA. CAU, so I go CAU. Careful on this one, there's 2 different amino acids here. So it's histine, HIS. And finally UAG, UAG, stop. So I'm going to use this chart here once again and find out what TYR, ALA, HIS amino acids, what protein that builds to build a protein to determine my eye color. So TYR, ALA, HIS. TYR, ALA, glue, right here. So TYR, ALA, HIS, orange and blue. Alright, and you just keep on going, and you go through until you get all 13 done, and when you're done with all 13, then you draw the monster that the DNA has coded for. And remember, you can get extra credit if you actually build a model, a good model, of the monster instead. Alright, good luck, and I will see you next Monday.